Topics in Post-Harvest Technology. Changes during ripening, Part 2. Carbohydrates, acids, and volatiles. Carbohydrates. The sugar content of many fruits increases during ripening. The exact blend of sugars and their concentrations varies from fruit to fruit. In some fruit, reducing sugars can accumulate. The two most common are fructose, also known as levulose, and glucose, also known as dextrose. The sugars accumulate in fruits such as apples, pears, and some varieties of watermelon. In other fruit, sucrose may be the dominant sugar. Sucrose is a disaccharide comprised of a glucose molecule bonded to a fructose molecule. Sucrose can occur at high levels in fruits such as ripe oranges, pineapples, and in certain varieties of watermelon. In fruits containing starch, sugar accumulation often correlates with starch degradation. Think of bananas, apples, and stone fruit. Starch is a polymer of glucose molecules connected by alpha linkages. Alpha linkages are easy to break enzymatically. Cellulose is also a polymer of glucose, but unlike starch, the molecules are connected by beta linkages, and these linkages are very difficult to break enzymatically. The sugar contents of ripe fruits can vary considerably. One may estimate the overall sugar content in plant tissues using a refractometer. Refraction is the bending of light as it moves from one medium to another. A refractometer measures the refraction of light caused by the total soluble solids in the plant cytoplasm. Total soluble solids refers to everything dissolved in the cell cytoplasm and may include sugars, proteins, and mineral ions. The higher the total soluble solids concentration in the tissues, the greater light is refracted as it enters the tissues. The bending of the light is calibrated to a scale, and we often use the degree brick scale in agriculture. The scale is named after Adolf Brix, who accurately measured the refraction of light caused by sucrose solutions of increasing concentration. Degree bricks correlate with sugar content, but do not indicate absolute sugar values since total soluble solids is what is being actually measured. However, this is not a practical issue for the ag industry since the majority of soluble solids in tissues are often sugars. Think of grapes, stone fruit, and melons. Here is a labeled photo of a handheld analog field refractometer. Analog simply means it's not digital or battery powered. In this case, sunlight passing through the prism allows us to take readings. The refractometer has a prismatic surface. This is where you would put a drop or two of juice gently squeezed from the fruit. There is a cover that comes down, and when you lower the cover, you want to do so gently so as to exclude as much air as possible, as air bubbles can prevent one from getting a clear reading. There's an eyepiece and a focusing ring, which allows one to focus the sharpness of the internal degree brick scale. This image shows the refractometer with the cover down. Imagine that a drop or two of fruit juice has been placed on the prism before the cover was lowered. You then hold the eyepiece up to your eye. Sunlight entering the prismatic surface before traveling down the tube is bent depending on the amount of total soluble solids in the fruit juice, and the sunlight causes an interface to form on the brick scale in the eyepiece. When you look through the eyepiece, this is what you will see. There's a blue zone and a clear zone, and depending on the concentration of total soluble solids in the liquid, the interface between the blue and the clear zones will move up or down the brick scale. In this case, when we read the scale, we're able to read approximately 20.5 degree bricks in the solution, or 20.5% total soluble solids. Notice at the bottom of the scale is written degree bricks, 20 degrees centigrade. 20 degrees centigrade is 68 degrees Fahrenheit. The brick scale is calibrated to accurately measure total soluble solids at 68 degrees Fahrenheit. Analog refractometers cannot compensate for temperature. When measuring at temperatures below 68 degrees Fahrenheit, the refractometer will overestimate total soluble solids. When measuring at temperatures above 68 degrees Fahrenheit, the refractometer will underestimate total soluble solids. If high accuracy is important, many digital field refractometers can measure and compensate for ambient temperature. Here is a lab or Abbe tabletop refractometer. It's more accurate than a handheld field refractometer, but it's also a lot more expensive. And in most cases, you're not going to bring this type of an instrument out into the field. Notice that it has an electrical cord. The typical cost for a good quality analog field refractometer will be in the $125 to $200 range. A good digital field refractometer will cost $200 to $400. A high accuracy Abbe tabletop refractometer will cost in the $4,000 to $7,000 range. Lemons do not contain much sugar. Lemon juice contains only about 1% sugar. However, refractometer readings will indicate that the typical lemon has between 8 and 9 degree bricks. That's because the refractometer is reading total soluble solids. The majority of solids dissolved in the lemon juice are acids, not sugar, so using a refractometer with lemons can lead to very erroneous conclusions about its sugar content. Sugar levels may increase or decrease during ripening. The exact pattern is dependent on plant species, variety, and the pre- and post-harvest environment. The rate of decrease or increase of sugar is very temperature dependent. Here we have two figures that indicate the changes that occur during the growth and ripening of pineapple fruit. The scale at the bottom is in days, beginning with the flowering of the pineapple plant. On this scale, 0 is the day that the fruit are ripe. Prior to ripening, the days are numbered negatively. After ripening, the days are numbered positively. When you observe this figure, you can see there are quite a few changes as the fruit grows and develops. With respect to ripening, let's look at the reducing sugars. We can see that as the fruit ripens and afterwards, the reducing sugars increase in concentration. Acidity. Flavor in fruits is comprised of sweetness and sourness in combination with volatiles. Sourness in fruits is primarily due to the accumulation of organic acids in the cells, and these are derived primarily from the Krebs cycle and some associated pathways. Within the Krebs cycle, or tricarboxylic acid cycle, we can find citrate, succinic acid, malic acid, and oxalic acid. Fruits may contain malic, citric, or tannic acids, tartaric acid, which is often found in grapes, and oxalic acid. The primary acids found in ripe fruit is species and variety dependent. In all fruits, acid content declines as the fruit ripens. The rate of loss is usually higher at higher temperatures. Volatiles. Volatiles are active in very minute quantities, in parts per million quantities or less. Many volatiles, and there are often over 100, are produced by ripening fruit, but usually only a few are involved in producing a fruit's characteristic scent. In general, Volatiles decrease once fruit have fully ripened and have begun to senesce. In climacteric fruit, volatile production coincides with the respiratory peak. Climacteric fruit produce ethylene autocatalytically prior and during ripening. This means that the ethylene triggers the production of more ethylene from the fruit. The ethylene triggers quick ripening, and the energy needed for this process is provided by an increased respiration rate, which we call the climacteric. Summary points. When ripening, fruits may undergo a number of changes, including changes in sweetness, acidity, and aroma. The sugar content of many fruits increases during ripening, and sugar as total soluble solids is measured using a refractometer. Acid levels in fruit decrease during ripening. The acids in fruit are primarily derived from the Krebs cycle and associated pathways. During ripening, volatile production often increases. The volatiles are active at very low levels in the range of parts per million or less. Though a fruit may produce over 100 volatiles when ripening, only a few contribute to the fruit's characteristic aroma and taste. Flavor is a combination of sweetness, sourness, and select volatiles.